Hello, this is Dr. Rina Rani Ray. I will be discussing about bacterial endospore today. An endospore is a dormant, tough, and non-reproductive structure produced by some bacteria in the phylum Firmicutes. The name endospore is suggestive of a spore or seed-like form, but it is not a true spore. It is a stripped down dormant form to which the bacterium can reduce itself. So here we can see the structure of the endospore. We can see the structures here also. Endospore formation is usually triggered by a lack of nutrients and usually occurs in gram-positive bacteria. In endospore formation, the bacterium divides within the cell wall and one side then engulfs the other. So here we can see the endospore in the bacterial cell. Endospores enable bacteria to lie dormant for extended periods, even centuries. There are many reports of spores remaining viable over 10,000 years, and revival of spores millions of years old has been claimed. When the environment becomes more favorable, the endospore can reactivate itself to the vegetative state. There is one report here. We can see the report of viable spore of Bacillus marismortieri in salt crystals approximately 250 million years old. So this is the oldest bacteria ever found. And here we can see the endospores. So most types of bacteria cannot change to the endospore form. Those who are able to form endospore are Bacillus cereus, Clostridium tetani, Bacillus anthracis, Clostridium botulinum, and Bacillus thuringiensis. Now regarding its chemical nature, the dipicolinic acid is a spore-specific chemical that appears to help in the ability for endospores to maintain dormancy. This chemical accounts for up to 10% of the spore's dry weight. So here we can see the chemical configuration of dipicolinic acid. The endospore consists of the bacterium's DNA, ribosomes, and large amount of dipicolinic acid. Endospores can survive without nutrients. They are resistant to different adverse conditions like ultraviolet radiation, desiccation, high temperature, extreme freezing, and chemical disinfectants. Common antibacterial agents that work by destroying vegetative cell walls do not actually affect endospores. Endospores are commonly found in soil and water where they may survive for long periods of time. A variety of different microorganisms form spores or cysts, but the endospores of low GC gram-positive bacteria are by far the most resistant to harsh conditions. So here we can see the structure of endospore. This is the endosporium, the outermost layer. This is a thin, delicate covering, followed by the spore coat. This is of several protein layers, thick, impermeable, which gives resistance. Then comes the cortex, which is made up of peptidoglycan, cross-linked with vegetative cell. It occupies about half of the spore volume. Then we can see the spore core, that is a normal cell structure, but metabolically inactive. And the core is surrounded by the core wall. And here we can see the core is containing DNA. So this is the spore mother cell, which contains the endospore. The spore mother cell is also known as the sporogonium. This is for protection. Core plays a major role in resistance with very low amount of water, slightly lower pH, high amount of dipicolinic acid complexed with calcium ions. Small acid-soluble DNA binding proteins are remaining present, which saturates DNA and protect them. There are different types of endospores, like central endospore, which is placed at the central position of the cell, then serve terminal, terminal, and terminal with solen sporangium. So here we can see 4 major types of spore distribution. Now we are coming to endospore formation. First of all, we can see the cell is dividing. So this is the cytoplasmic membrane and the cell wall. So after the replication of DNA, we can see the division of the cell. Now cellular division of cytoplasmic membrane, it leads to pre-spore formation begins when one septum arises here. And this is called the pre-spore. That is the prerequisite or the pre-spore position. And now it is forming the cortex, and the other part of the cell which contains the DNA, this DNA is getting denatured. Now we are concentrating on the spore. What happens? Let us see. So this is the spore coat, and then the spore is going to form gradually. Now maturation begins, exosporium formation. So another covering is there. So it is called exosporium. Now this mother cell releases, matures spore, and this spore is getting out from this mother body, and it is coming as a free spore. So here we can see also, this is the DNA, the sporogonium. Let us see what happens. The division is taking place. Now a pre-spore will be formed after the formation of a septum here. So it includes the DNA. This is the mother cell. Now we can see this is the spore is forming. Yeah, this is a pre-spore. Now the spore is being formed. This is the spore. And now this part of the DNA is getting denatured. And now the spore has been formed. Yeah, the spore coat has been formed. The cortex is there. Now the maturation takes place. Yeah. Now it is trying to go out from the spore mother body. Yeah, here we can see the mature spore with exosporium. Now the spore is going to release. Yeah, it is now released. It is a free spore. So again, we can see here. So this is the vegetative cell. Vegetative cell, now the pre-spore is being formed after the appearance of the septum, spore. Then the cortex formation occurs, the core formation, and the core coat is there. Now spore maturation. After spore maturation, the cell lyses and endospore releases. Now sporulation requires the presence of free oxygen. In the natural situation, the vegetative cycles occur within the low oxygen environment of the infected host and within the host. The organism is exclusively in the vegetative form. Once outside the host, sporulation commences upon exposure to the air, and the spore forms are essentially the exclusive phase in the environment. The spore formation, that is sporogenesis or sporulation, normally commences when the growth ceases due to lack of nutrients and other adverse conditions. It is a complex process and may be divided into 7 stages. The stage 1, that is an axial filament of nuclear material, forms, followed by an inward folding of the cell membrane to enclose part of the DNA and produce a 4-spore septum, that is the stage 2. Now comes stage 3, when the membrane continues to grow and engulfs the immature spore in a second membrane. Next, the cortex is laid down in the space between the two membranes, and both calcium and dipicolinic acid are accumulated. That is our stage 4. Finally, lytic enzymes destroy the sporangium, releasing the spore. This is our spore stage 7. And in between 4 and 7, the two stages, they are for maturation. Now, the endospore germinates and activates. So here we can see the spore, the endospore. Now, after activation, this is the stage 1, cation releases, calcium dipicolinic acid release, partial co-hydration, and some loss of resistance. Now the spore germinates. This is stage 2, that is cortex hydrolysis. Further core hydration, that means more water molecules are accumulated, core expansion, more loss of resistance and loss of dormancy. And finally, the outgrowth, that is metabolism, then SAS degradation, macromolecular synthesis, and ultimately escape from the spore coats. So this is the coat remnants, and it is now going to germinate as a vegetative body. The transformation of dormant spores into active vegetative cells seems almost as complex a process as sporogenesis. So 3 processes are there: activation, germination, and outgrowth. Endospore will not germinate successfully even in a nutrient-rich medium unless it has been activated. Activation is a reversible process that prepares spores for germination and usually results from treatments like heating. It is followed by germination. The breaking of spores' dormant state or the activation this process is characterized by spore swelling, rupture or absorption of the spore coat, loss of resistance to heat and other stresses, loss of refractivity, release of spore components, and increase in metabolic activity. Many normal metabolites or nutrients like amino acids and sugars can trigger germination after activation. Germination is followed by the 3rd stage, that is outgrowth. The spore protoplast makes new components, emerges from the remnants of the spore coat, and develops again into an active bacterium. With a simplified model for cellular differentiation, the molecular details of endospore formation have been extensively studied, specifically in the model organism Bacillus subtilis. As we know, endospore-producing bacteria are dangerous pathogens. So like endospores of the bacterium Bacillus anthracis were used in 2001, and it is known as anthrax attacks. The powder actually found in contaminated postal letters consisted of anthrax endospores. This international distribution led to 22 known cases of anthrax, 11 inhalation and 11 cutaneous. The case fatality rate among those patients with inhalation of anthrax spores was 45%. The 6 other individuals with inhalation anthrax and all the individuals with cutaneous anthrax were recovered. Now, apart from this biological warfare, let us concentrate on the positive aspects of the spores, the application. The food spoilage endospores producing bacteria has become a major quality target organism. The spore coat can be used as a vehicle for heterogeneous antigen presentation and protective immunization or vaccination. As interest in the spore coat increases, it seems likely that they will be exploited further for drug and enzyme delivery, as well as a source of novel self-assembling proteins. Bacterial endospores can serve as fast genome protection shells against various environmental stresses to enhance microbial control applications. So this is all about the endospore, bacterial endospore. And with that, I would like to conclude. Thank you.